Every single second, your body is generating electricity. You can actually see it happening here, plotted out on the jagged output line of a standard electrocardiogram. Up in the brain, millions of neurons are firing off so much collective electrical energy that we can track it using wires placed directly on the surface of the skin. Humans don't have copper wires or lithium-ion batteries inside them. We are squishy organisms made primarily of water, fat, and salt. So how exactly are we powering these signals? To find the power source, we have to zoom way down to the microscopic level, where the cell operates as a complex electrical assembly. By translating biology into these engineering principles, we can build mathematical models that explain how these tiny currents drive our thoughts and heartbeats. The boundary of every cell is a membrane. It's a wall made of 2 layers of lipids, or fats, and it is incredibly thin, measuring only about 5 nanometers across. Because fat repels water, this membrane acts as a perfect electrical insulator. It functions exactly like an electronic capacitor, separating charged particles to create a positive charge on the outside and a negative charge lining the inside wall. To build that charge, specialized pumps actively push sodium ions out while pulling potassium ions inside. That resulting electrical imbalance creates a steady baseline voltage across the membrane. We call this the resting membrane potential, and in a typical nerve cell, it hovers right at -70 millivolts. Scientists can calculate this exact voltage using mathematical models like the Nernst and Goldman equations, which factor in the precise concentrations of those sodium and potassium ions. Because of this constant pumping and separating of charge, every resting cell is effectively a fully charged battery, coiled and waiting to be unleashed. Embedded throughout that fatty membrane are specialized protein nanopores called ion channels. In circuit terms, these act as highly selective variable resistors, or electrical switches. These biological switches are firmly clamped shut. They only pop open when struck by a specific trigger, like a sudden shift in nearby voltage or the arrival of a chemical messenger. When that trigger hits, we get a spark. Look at this graph plotting the cell's internal voltage over time. As the trigger occurs, the sodium switches fly open. Positive sodium ions rush inside, and the internal voltage violently spikes from -70 up to +30 millivolts. This spike is known as the action potential. The reset happens immediately. A different set of switches opens, these ones specifically for potassium. They open a bit more sluggishly, flushing positive ions back out of the cell, dropping the voltage rapidly back down before it levels off to our starting baseline. In the 1950s, researchers like Alan Hodgkin mapped this exact current flow, creating a quantitative mathematical model that correctly predicted how these ion channels operate. This rapid, millisecond-long chain reaction of opening and closing switches is the fundamental all-or-nothing unit of communication for your entire nervous system. A single biological spark doesn't stay trapped in one place. As one patch of the membrane spikes in voltage, it triggers the neighboring switches to open, sending a continuous electrical pulse traveling rapidly down the length of the nerve fiber. The duration of this electrical wave physically changes depending on the job the biological hardware is performing. This side-by-side chart shows how those signals adapt. On the left is the fast 1 millisecond firing of a neuron, built for rapid thought. On the right, a heart muscle cell holds its electrical spark open for 300 milliseconds, creating a wide plateau. The physical shape and timing of that electrical wave dictates what the organ can do. A narrow spike lets you recall a memory, while a prolonged plateau ensures your heart maintains a steady, rhythmic beat. Sometimes this microscopic grid fails. Genetic mutations can warp the shape of the ion channels, causing them to stay open slightly too long and delaying the critical electrical reset. When this delay happens in cardiac tissue, it causes a condition called long QT syndrome. The heart's rhythm falls out of sync, creating the potential for lethal electrical short circuits. To fix problems like this, engineers use specialized tools, like these microscopic glass pipettes, to measure faulty currents passing through a single ion channel. Biomedical engineers take that precise cellular data and use it to calibrate medical hardware. It tells them exactly how to design pacemakers, configure diagnostic monitors, and build neurostimulators. Mastering this bioelectric code provides the blueprint for engineering targeted interventions that synchronize with the body's native electrical rhythm.